It's a great pleasure to be here in this virtual CAMDA conference, and I would like to present the summary of our recent result or the work, what we did in these unusual circumstances. And my name is Paweł Łabaj. I'm working at Małopolska Center of Biotechnology at Jagiellonian University in Krakow, and I'm going to tell you today a bit about the DNA-based methods in intelligence and how we are moving towards metagenomics. We are at the CAMDA conference, which focuses on one of the grand challenges of our time, the intelligent exploitation of the big data in life sciences. And as you know, the CAMDA offers multiple very challenging and very unusual data sets for the many years, around 20 years already, and in not only about the cancer on data integration, but in the past few years also about investigating the environmental metagenomic data. And this would not be possible without collaboration of CAMDA with the MetaSAP, where in MetaSAP we aim to build a molecular profile of the cities around the globe to improve their design, functionality, and the impact of the health. And we are doing this by collecting environmental samples mostly in the metro systems. But how it all starts? So Chris Mason from New York and his team started collecting the swabs in the metro system in New York. And once they had enough, they decided, okay, let's see what we can find. And what has appeared? It appeared that about half of the reads which were sequenced cannot be mapped to anything what we know. But what also was interesting that what we can identify, in fact, is harmless, and we don't have to be worried about uh, traveling with the metro system. Of course, in the current situation with the SARS virus, it's not that easy. But in principle, when there's no pandemic, the metro system and any other public transport system is safe. And safe to this extent that Chris has said once that he would advise any new parent to roll their child on the floor of the New York subway. Because even if we are able to find some pathogens, they are on very low quantity, which means that this dust from the metro system or any other public system could be treated, in fact, as a very complex vaccination, providing a huge variety of different pathogens, but in very, very low quantities. So once it has started, and there were the results from the New York, the question was, okay, we know what lives in the New York metro system, but what about other cities? What about the Tokyo, London, Berlin, Hong Kong, and all the other locations, the big metropolis around the world? And this was the time when the concept of MetaSAP has born. So the MetaSAP is the metagenomic and meta design of the subways and urban biomes, and it has been launched in 2015. And since then, it's evolving quite rapidly, and we have many different interesting projects. But we have started with this manuscript about what we are really aiming for, because what we have realized quite quickly is that there is totally no standards about how to collect samples, how to process them, how to analyze them when we are talking about the whole metagenomic sequencing of environmental samples. So this was the goal of the MetaSub. And the main project which we are pursuing within the MetaSub is something what we call Global City Sampling Day. And within this Global City Sampling Day, at the one specific time point, most cases this is 21st of June, we collect the environmental samples in the public transportation system around the world. At the moment, in Metasap, we have around 100 cities from all continents, from all the world, and we are collecting the samples once per year in the very centralized and synchronized way. As you can see here on the figure, the highest peak is around the 21st of June, but of course, depending on the different circumstances in the different regions, areas, cities, it's taking a bit of it can take a bit longer. In this year, it will be prolonged because due to the pandemic situation, not in every city were able to get permission on time. Sometimes there are some additional requirements we need to fulfill, But in general, yes, this year we are also doing the sampling of within the Global City Sampling Day. So as you can see, this is a global effort, and we hope that having this data, we can really provide a new insight to what lives around us, and that this knowledge will be very useful in terms of our health, but also the planning of the city. Right now in the revision, there is a manuscript which has been prepared by the MetaSub consortium and which is summarizing the first few years of the Global City Sampling Day. So we analyzed the thousands of samples, and we are looking in new insight and creating the global genetic cartography on urban metagenomes and antimicrobial resistance, because this is really what we are aiming for. At the beginning of the MetaSub, we were asked many times, okay, it's very interesting. It could be something like a master project for the maybe citizen science, but where is the real science there? And in the beginning, we were not able really to say what is the main focus, because yes, it was a data exploration effort. But now we can see that we can track the antimicrobial resistance, but we also make something like a global map of the urban microbiome and to see how it's changing over the time. So this manuscript is already available on the bioRxiv, and it's undergoing the revision, and we hope that soon it will be published, so you'll be able to get it in the final form, but already now it's available to download and to read, and maybe to provide some feedback to us what else we can do and what else might be interesting, or maybe you would like to also join MetaSub and our efforts. But having this data from Global City Sampling Day was really a very interesting and unusual situation that we can put and take this data and construct a CAMDA challenges, because nobody before was trying to do something like this. So at the beginning in 2017, we were curious whether if we have read the data from different cities, whether we can differentiate between the cities based on the molecular profiles, and if yes, what can be used? Maybe bacteria, maybe plants, maybe something from animals. So it was the first challenge. We call it MetaSub Intercity Challenge because we had the 3 cities from the US, which were we had samples which were presenting the urban microbiomes of these cities, and it was a data exploration challenge to compare organism fingerprints from the public places across cities and investigate organism sequencing and biodiversity in context of the location. So the output was this: yes, we can differentiate the cities. And once it was clear that we can do this, we came back next year and provide from MetaSub more data. So in 2018, we have hundreds of novel city microbiome profiles. And the question is, okay, we will provide a training set, but also later the testing set. And the question would be whether we can properly classify, or even maybe we can predict from where the mystery samples are coming from. And it appeared that yes, classification can be done quite accurately, even with having just the hundreds of samples. But with the prediction of the novel samples, where they are really coming from, it was very, very difficult, because there were not enough data which could provide the necessary information. That's why in CAMDA 2019 we extended the dataset. First of all, we provided thousands of the samples from the MetaSub, but we wanted that participants will study it in the context of the other data which are available, like from the Earth Microbiome Project or anything which can be used as well. So here, this challenge was similar. I mean, we wanted first to learn whether the classification can be improved and whether it would be possible really to predict where the new samples are coming from. But it appeared again that predicting from where the mystery samples are coming from is still too challenging. And what was pointed out by the participants was that we might need to think about the Earth as a multiple locations of the specific ecological niches, where the climate, but also any other things, including the soil composition, might affect what really is living there. That is why this year in CAMDA 2020 we have added such information, and I hope that we got way better results than it was in the past. But this uh, I cannot say at the moment because I'm pre-recording this talk, so I don't know what was the final results of the participants. Anyhow, this was gave us a foundation for thinking that we can really use the metagenomic samples for forensic. And taking together the expertise and experience from these uh, three CAMDA challenges and taking into account also the authors who are working for the forensics, we decided to write a perspective review paper, which is already available in the preprints archive and is also undergoing the submission into the journal. And again, we hope that it soon will be available. You can see that it already brought some attention. We have more than 600 views and over 300 downloads. It means that really such summary, such review and perspective, it's really needed because there is still a lot of to do. So where it comes from and why we think that it might be really useful is that they're already at the same, more or less at the same time when we were investigating these issues with the CAMDA, appeared a set of papers which were showing that it really should be possible. So first of all, in 2018, there was published a global atlas of the dominant bacteria, which we see that yes, there is a group of bacteria which can be seen almost all over the world, but there's still some set of bacteria which are characteristic from the different locations. So if they are characteristic for different locations, continents, or maybe some smaller regions, it should be possible to do the prediction of the sample origin based on the metagenomic profiles. Especially that we can see that it's highly connected with the climate or also the soil and all other conditions like the pH cluster, dry lands or the low productivity clusters. So all these uh, factors are influencing of creating some specific ecological niche, which is then occupied with a specific set of the bacteria, specific community. So if we can decode or unveil what bacteria and what communities are living in a specific ecological niches, it would make possible to create a global map which could be used for the sample origin prediction. So this would be our ultimate. And looking also into the other papers where the scientists from Denmark were studying the sewages, you can also see that there are some characteristic patterns, both on the different levels, on the antimicrobial resistance classes, antimicrobial resistance genes. So you can see that there are some specific groups of bacteria sharing these genes and classes, which can be found higher on the higher abundance or lower abundance in different regions all over the world. And in what was interesting that using the antimicrobial resistance genes as a markers for the mystery samples, specifically from the urban microbiome, was used by one team in 2018, and this team has obtained very good results in prediction in classification from of the samples. So I think this is really the functional approach. It's one of the very prominent and I think very the approach which has a great perspectives in this. And with the Nematosap, we also did some kind of such testing and prediction, and we also obtained similarly result about 80-90% as it was done by the CAMDA participants. So we can see that it doesn't matter who's trying to play with this concept and with this challenge, either the CAMDA participants or the Metasap consortium members. Yes, it can be done, and we can, and thus we can think that we are at the beginning of the road where we can provide the tools which can be later used in intelligence and forensics. Because how it can be helpful when we talk with the scientists which normally are working with the forensic, they say, okay, so whether the microbiome, the metagenomic DNA can be helpful when there's no human DNA at the crime scene. And in fact, there are several approaches where you can say, yes, it can be useful. Already now it is used for the reducing the suspect pool because we know that there is characteristic microbiome of a person. It can also help to accurately predict the time of death and help to elucidate the cause of death. So this is what is already in use. But what we are aiming for is to give a hint toward geographical localization of the individual or item. But there are still some issues we need to solve because in principle, the method works. So we have a proof of concept, but to make it ready for forensic, there are still some road which we need to follow. So the issues is that we need to really, all the sampling, sample treatment, and DNA extraction procedures, they strongly influence the quality of the recovered metagenomic DNA. So we need to standardize it. We need to have consistent sampling procedures at the crime site, so each time it will be done exactly the same way. We need to also minimize and control for the background DNA from unwanted sources, like the laboratory environment or contamination through consumables and reagents, which sometimes is called as a ketome. So we need to try to control it, prepare the standard operating procedures, and validate them. We also need to think whether we need to do the whole metagenome sequencing or maybe we should rather focus on some specific area and make the marker selection. And of course, we need to investigate and improve further the bioinformatics analysis stability, because so far it was shown that depending which tools we are using, on the same data we might get a slightly or even not slightly different results. And in the end, we also need to think what are the ethical considerations in all this. In summary, the whole genome and targeted metagenomics are showing the benefits and potentials for forensic and intelligence. However, there are still challenges we need to address. So there is a need for standard operating procedures. We need to rigorous validation that each analytical method and step of the process. And we need to create a reliable and comprehensive database and references to accurately interpret the obtained results. So I would like to thank the CAMDA organizer and participants, the MetaSAB consortium members, as well as the members of the Małopolska Center of Biotechnology, of my lab and lab of Professor Barnicki. Thank you for your attention.